שלום לכולם. מה שלומכם?
טוב, תודה. מה שלומך?
בסדר. אוקיי. אני אתחיל. One update, I cannot put weight on my leg for the next 6 weeks. So I'm not coming to the university, unfortunately. So we'll continue this mode of remote teaching till the end of the semester. Maybe we'll be fortunate and we can have, I don't know, maybe the last session we can do on site or something like that, but the rest, I'm not going anywhere. We're starting today with the first student lecture. Anatoly is here? Well, he should be here because he starts, oh, here he is joining us, because he's starting relatively early. I mean, he is, so the way that we did it, I think it is working pretty well so far, the way that we organize these student talks. I mean, last year it was a disaster because students spoke for like half an hour, 40 minutes. It took all the time of the lesson, so I couldn't teach anyway. I'm feeling that I don't have enough time to show you all the cool stuff that I want to show you. And with that, I had even less. And it was it was not focused. It was not, I mean, the student was speaking and most of the students, it wasn't to the point. It was like, I mean, I think that some guidance is needed, especially because the topic is so interdisciplinary and so it's not simple, right? I mean, it's hard to understand what is key and what is a side. And so I had a very good experience so far with this two-step preparation of Yeshaya and then me. But maybe it's because I had Anatoly as our first victim and he's fantastic. So, I mean, you have to stand up for high standards now. Anyway, so we'll continue with that. We have, I think, for the deep learning parts of at least the first deep learning in this context of mapping between one domain and another, we have the talks are going to be this week, next week, and the following week. And I think that that's it. I'm not committed for that, but I think that will be enough for this topic. So if you want to present a paper on this topic, then hurry up. I am going to bring up topics as we go, based on the topics that we speak in class. Any questions, concerns, something else administrative? Okay. So I'm starting. You can see my screen, right? Beautiful. Adi, I will appreciate לפתוח מצלמות. Otherwise, I mean, you can watch the recording. I really want people to be here, to be involved, to actually, I want to see people that I speak to, although it's from home. ואלון גם כן. Okay. So here are the topics that we are discussing and going to discuss. Image segmentation is one topic that I didn't include in the points that I showed before. This is one of the classic computer vision, and especially in microscopy, segmentation is one of the most basic tasks that we need to do. And deep learning also have done a big contribution to segmentation and has revolutionized our ability to segment cells. It's not only that, because that was true also a year ago, and I decided not to include that in the course. But I think that a few recent papers that came in the last year and a half have really showed other cool aspects that we can learn something from, not just showing better segmentation, but some things that I think go beyond that. So I decided to include it this year. And I'll probably give like a short introduction, but we have a few students' presentations about that in 2 weeks from now. Enhancing cell image quality with deep learning, I showed you a few examples last time that we met, and we're going to see a few more today. And we're going to continue what we just started in 2 weeks ago, talking about generative model for cell structure and classifying cell state with deep learning. Probably we're not going to finish everything that I have in this presentation, so it will go a little bit into next week. And after that, I think that we can move on to other topics, exciting topics. Not everything is just deep learning and microscopy. So a quick reminder, this is the figure that I really like, a really nice explanation of the CARE paper. So I remind you, we have trade-offs, right? Trade-offs between the ability to have a good signal to noise on one hand, and killing ourselves with laser power on the other hand, imaging fast and imaging a large field of view. Everything, when we go to one direction, we pay in another direction. And of course, we want to have it all. So CARE showed one nice trick, and they did it really nice. I really love the ways that they approach that and all the thoroughness. They showed it on a lot of data sets. They did many controls and many from very different angles. They came and tried to test it from many directions. It was very detailed and through. So I think, and it's actually a method that actually works. So I think it's a very good example, and I'm not sorry that I spent on it more than an hour, I think. Anyway, the idea is very simple. The idea is we have a high quality image here at the top, a low quality image at the bottom, and we learn a mapping between the low quality image to the high quality image using a generative neural network. And then once we have this mapping, we can image in conditions that are of low quality and generate images of high quality. That's the idea. That's all. One of the nice things in their papers, they showed the 3 layers of being able to do that. One was actually directly taking matched pairs of high resolution and low resolution images. The second was taking taking images but simulating from the image low quality images, low resolution images. They did that by looking at 2 dimensions and simulating the 3rd dimension. And the 3rd example, which was cool, was taking completely simulated data and using it in order to train and then artificially deteriorating the quality of the image, and then training a network to learn the reverse mapping, and then actually shows that they can actually use it on real data, on experimental data, which was I think was really cool. The architecture was a UNet, which we'll see again and again here in this course. And just one movie as a reminder. So this is the raw image quality, what you see, and what we see, what we go here and see the scanning is the computational restoration. And if our goal is to actually identify the single cells and track them over time, then you can see that in the original image, it's really hard. And in the image after this correction, it's much easier to actually identify the single object within it and to track them. The second example was Anna Palm. the task there, which came a little bit earlier than CARE, if you want to be accurate in the credit race. It's not really. I mean, both of them are cool, and both of them I really like them. I think that CARE is more methodologically more complete, so this is why I highlighted that. But I really like Anapalm as well. And the idea there is that we can take, instead of in super resolution, instead of taking thousands of images, 10,000s of images, or a million images of diffraction limited spots that are stochastically emitting, instead of that, we can take a much smaller subsample and then learn the mapping from a low quality localization image to a high quality localization image. And I mean, I'm not going to go into the details here again. I think that the idea is cool. So taking just a short, taking data that was generated with a short time scale and then using it in order to compensate for the large time scale of many of taking a much more images. And eventually this is important again for the ability to image faster. So super resolution is usually done for fixed images, so you don't have time information. But if we can do it, or very low resolution in time, but if we do that with much smaller number of images, then we can start doing live imaging in temporal resolutions that make sense in the biological scale. One thing that maybe I do want to mention is that they showed that they can take the wide field image, which is not even, there is nothing about super resolution there, but they can learn a mapping from the wide field into the super resolved image. So the wide field is what you see here on the left, the low quality image, and learn a mapping to the high quality localization image. So from here, you can see my akhbar, right? Okay. So from here to here, and it does most of the job it does by itself, right? So you get most of the structure you get from is hidden, is encapsulated within the wide field image. But on top of that, when we add the localization, we get all the fine details much better. And I think this is a nice contribution of showing what you can get from these two different modalities of the localization images, a small number of localization images, and the wide field together. Okay, and now I'm turning the stage to our guest lecture for today, Anatoly, soon to be Dr. Anatoly Shusterman. Right? When are you graduating? And Anatoly, אתה איתנו? Anatoly. Okay, he went to the worst time probably, but okay. So I'll say a few words about this paper, and if he's not here yet, then I'll go to the next paper, which is related. Anatoly, אתה איתנו? חשוב מאוד, כי עכשיו זה שלך, עכשיו it's your turn now to speak. So Anatoly is going to talk about the Deep Storm, which is a paper that came out also early 2018. It's also if you think about it in the timeline, it's very close to the Anapalm paper, and also the task is quite similar. So this is super resolution also, super resolution single molecule microscopy using deep learning to the Anapalm and to the CARE papers. And it's a paper, and there is another reason why this paper is cool, and I'm happy that Anatoly picked it, because we have here local pride in Yoav Shechtman from the Technion, who is of course an Israeli, and that's really cool stuff. So we're going to hear Anatoly speak about this paper, and then I'm going to speak about the follow-up paper that came out last year from Yoav's group. Anatoly, the stage is yours.
Okay.
I'm starting timing you.
Okay. Let's share. Do you see that? Great. Okay, so I'll talk today about deep storm super resolution single molecule microscopy by deep learning. Before we start talking about the paper, let's discuss the topic of localization in microscopy and why does it needed. Localization in microscopy is a task of location of molecules in wide field images. The complexity in this task is separating dense emitters in high resolution. We can see an example of structural analysis. In image A, you can see the wide field image of dense emitters. From this image, you cannot separate between small emitters. When you apply palm technique, which is a photoactivated localization microscopy, you can see in B high resolved image, which specialists can analyze in its molecular structure. Super resolution takes big parts in localization tasks, as a raw wide field dense emitter image cannot be analyzed. But we have lots of algorithms that does this job. So why do we need another one? Although lots of algorithms exist, they have several limitations. The data processing time requires lots of time, thus it's not scalable. It requires exhausting parameter tuning for sample and not for a domain, makes these algorithms usable by domain experts only. Also, these algorithms require knowledge of the general sample structure, which makes them hard to use. In Deep Storm paper introduces a precise fast parameter-free super resolution image reconstruction method using deep learning model. In this paper, like in Anapalm, a paper they wanted to speed up the processing speed of the data, this model is capable of reconstructing dense emitters wide field image in A to a super resolved image in B. You can see it in B that you can identify single emitters, though in A you cannot do this. Deep Storm uses a convolutional autoencoder model to reconstruct the wide field image to a super resolved image. Autoencoder is a neural network which compresses the image and it's trained to reconstruct the same image in the output. The autoencoder network middle layer is a bottleneck which controls the compression level. When the size of the bottleneck is greater, it compresses less, and in the extreme case, it can contain and learn the whole input. When the bottleneck size is lower, it's used as a lossy compressor, like a JPEG in photos or MP3 in audio. Ultimately, the input is compressed as much as it loses the noise, and it's able to reconstruct the input, preserving the logic of the data domain. In Deep Storm, they take the idea of the autoencoder, but instead of reconstruction the same image, in the input they have the raw input, and the output is a super resolved image. So how did they train the network? First, they took a dataset with super resolved dense images. Then they applied downsampling on the image to create the model input. The model training was applied in the opposite way. So the input was the blurry image, and the model target was super resolved image. In this way, the model trained to get the blurry input and to reconstruct the original image. So while estimating the autoencoder limits, the researchers first tried to estimate the expected resolution might be obtained from this autoencoder. They simulated strips with small grid of 12 nanometers and applied noise on it. You can see in figure B, number 1 to 10, 1 to 9 is the emitter density. 1 is the sparser and 9 is denser, which is hard to identify. Their purpose was to locate all the emitters. In C, we can see the distances between the strips. As the strip width went lower, the distances between them went lower too. This figure shows the reconstruction result. You have here the ground truth, and for each of the density, you have the reconstruction result. When analyzing the predicted strips in figure E, we can see that even with gap distances of 6 nanometers, the model output might distinguish between strips. Then the researchers compared the results obtained by Cell Zero algorithm versus the result obtained by DeepStorm. You have here the input and here the reconstruction of two algorithms. To achieve maximal result on Cell Zero, the researchers had to choose carefully the parameters of Cell Zero by error and trial. The usage of a DeepStorm network was out of the box without the need of hyperparameter tuning. When we magnify the green image, we can see in both algorithms a reconstruction of the blurry image, but DeepStorm is more detailed, and you have here more sharp strips, and here you have some false positives results. The DeepStorm reconstruction error rate was about half of the leading Cell Zero algorithm and stand for 37% versus 69% reconstruction error rate. Here we can see again the leverage of DeepStorm in reconstruction images, but now they applied it on simulated data. In the red box, we can see that DeepStorm algorithm was able to reconstruct the blurry image to an image that is closer to the ground truth, and it has less false positives like here in Cell Zero. That means that synthetic data generation might serve as a feed of lower performance model or to augment the experiment results. If you remember, here you have the results of 81% of true positives versus no, it's wrong. You have here true positives of 88 and the experimental is 96%. It's transposed. If you remember the CARE paper, that makes the ability to expose the animal to less laser rays and prevent it from suffering when you train it on simulated data. So when we compare the computational timers algorithm, we can see also the DeepStorm has leverage on other algorithms, especially with available GPU. If you have here the 4 or 27 seconds versus the large numbers here. So it's much faster. So in summary, we have a super resolution method comparable or better to the state of the art. We have a DeepStorm that is faster, lots faster than the lead algorithms, and you don't have to do parameter tuning, so you don't have to be an expert for that. And also, usually I'm very criticizing of the data science application in non-core data science papers, like in a cybersecurity domain that I'm researching. They tend to have mistakes with the metrics and with the methods, and sometimes they have problems with the reproducibility. So I couldn't find anything suspicious or fishy in this paper. It's a good paper, and that's it. Thanks. I have 5 seconds more.
Okay, questions?
the big difference between this algorithm and the other algorithms that we studied before, I'm not sure that I can understand what is the big difference, because the main concept is pretty much the same, right?
A reconstruction of the image. Yeah,
Yes,
they have
reconstruction and
same target. They
it's
have
both.
the same target. Yes.
It's both reconstruction and both are using autoencoders. So more or less the same. So what is the main thing here that makes it special?
Actually, I don't remember the previous. I didn't read it. Maybe Assaf can tell. I don't know.
Which one? You mean the Anna Palm paper?
It's actually in CARE also there are things like that. I mean, it's more or less the same. It's doing pretty much the same job. Isn't there any comparison between these and the previous?
No. So first they came in. So Anatoly, do you have anything to say before I?
No, I don't have. I don't remember this paper. The other one.
Okay. So so first think about the timing. I mean, they came in parallel, right? I mean, there are like a few papers that came in the same time. The Kerr paper, this paper, and the Anna Palm paper all came, a few months apart from one another. So they were in parallel. Actually, I remember that I think when I think about it, I think it was while I was interviewing for jobs in the Israeli Academy, and I was interviewing also in Yoav Shechtman department. He's in biomedical engineering at the Technion. And I think I told him about the CARE preprints that just came out. And he became a little bit, he was a little bit worried until he wanted to check exactly what they did and what, how much. So this is one thing that I want to say. So it came all together and this makes sense. But the other thing that maybe I'm recorded, so it's not very good, but I'm going to say it anyway. I mean, the same idea, you're going to see in some of the papers that you guys present as well. We're going to see it again and again in very top and prestigious journals for slightly different applications and different way of showing that, which I don't have a good explanation for that. I mean, besides that deep learning is cool, it just came to microscopy, so it has like 2 years of every paper coming in very high impact. In this case, the uniqueness is focusing on super resolution microscopy. The Anna Palm, the focus was on the idea that you can take much less images in time, right? Much less photo-activated, right, diffraction limited spots, images of that. Yeah. But they did show it implicitly because they used the wide field image, right? And they showed that the wide field image you can do most of the information you can extract from there. Here the reliance on simulated data was more. So yeah, I agree that it has a lot of very close aspect to the other project. One of the things another thing that comes to my mind now is that it is important. I mean, it's also some papers, some journals talk about it in terms of scooping. It is important when similar things come at the same time, and one of them validates the other. Yeah, so okay. You make sure that there is no one team who was lucky with some data set or cheated a little bit, or so when you get something and you see it coming around the same time from different groups, it makes you believe that it actually could work. So the journals also understand that, and they're trying to I assume trying to fit along. So okay. I think this is my interpretation, but I think it's a very good point.
Thanks.
Any other questions? Okay, so I just remembered, and I wanted to show that. So I told you I was looking for that, but I was trying to listen also, so I couldn't do it effectively. Now I'm trying to talk to you without doing that in parallel, so it's also not very effective. Oh, here it is, probably. 1 second. So what I wanted to show, because I remember that just when Anatoly spoke, is that no. No, I can't find it. Anyway, I told you guys about the zero cost DL for MIC, one of the platforms that makes deep learning available for the community of cell biologists to use. So they can, a cloud platform, they can just upload their data and use existing models for different tasks. So I actually noticed, I now I failed to find it, but I noticed that one of the models that they implemented there was exactly that, Deep Storm from Yoav Shechtman group. So now, in principle, people can use it. It's available for people to actually use, and it is something that is useful. I mean, using these filamentous structures, and I mean, it's something that you see a lot in cell biology, in microscopy. Okay.
Assaf, can I ask a question?
Yes.
Thank you. Do we use this deep storm and care in industry? Because there's always a very, very little chance that there'll be some error by default. So do we use it in industry?
I don't know, and I'm not aware of applications of super resolution in industry. So, I mean, super resolution is looking at very fine details. It's very high resolution within your cells. I'm not aware of industrial applications for that yet.
Do we trust it like 100% that it will be accurate, or do we still fear an error?
No, of course it's not. Of course it's not. I mean, we talked about the hallucination effect is still there. Of course, we cannot completely trust it. And another thing that we need to take into consideration, because it's young, the field is young, and these technologies are young, is that people did not systematically test that. People did not systematically test what you can, how much you can count on the output of these fake images, right, that are generated. What can you take that? What can you use it for, and what you cannot use it for? When are you going to say that you are not sure about your results? For now, it depends, and there is, every time when someone uses that, they need to be very careful about the validations. And frankly, most of the papers now are methods papers, so just you need to show that it does something cool, and you need to show, you can show that it has its limitations, and that's it. When it will grow as a field, and people will start using it to actually make biological discoveries, then we'll get into the point of can we really trust it and how much. And my lab we have one project that deals not with super resolution, but with other in silico labeling. So the things that I showed you 2 weeks ago toward the end of the class, and I'm going to show today to complete today, we are working about what are the right measurements, how can you trust the in silico, what can you use it for and what you cannot, and how to really make it something that is actually useful for biology. But that's another story.
No, thank you.
Okay, so Davitali, I think it was a very high standard, and now after you saw how it should be done, you should also be prepared. Okay, so I was happy that Anatoly selected this paper, because it made me read the follow-up paper, which is Deep Storm 3D, from the same group, a paper that came just last year, so less than a year ago, from Yoav Shechtman lab. it's basically a very similar idea of this mapping in terms of computation, but it has one aspect that I think makes it really, two aspects for you, because one of them you also don't know. So I think two aspects that makes it really cool. And the idea here, if you think about it very broadly, the idea here is to learn two things in parallel. One is the actual optical system parameters. So how are you going to acquire the image? Parameters of acquisition of the image. And also optimize the localization algorithm, and do that at the same time. And if we do it at the same time, we're going to get better microscopy, better optics, and that is optimized also in terms of the algorithm for localization. Sounds cool, right? Okay, so I mean, I'm not going to go over the components here, but you do know what is a point spread function, right? I told you about that, and Natalie told you about that, and we showed a few examples. So, but what is point spread function engineering? So point spread function engineering, and this is also Yoav Shachman is the world leader in that, is the ability to, so it's actually using like a filter, but a physical filter, designing a physical filter that will allow us to get a pattern that will give us information about the 3D position of a point in your image. So you get this type of these crazy phase patterns, it's called. There are other names for that, but phase patterns that are used to actually take a point object and transform it to some object that give you information about the location in 3D. Okay? So if you look at without this phase mask, you're going to get a PSF, point spread function, and this depends on the location in 3D, right? That you're at. And you can see, I mean, it looks like a PSF, right? You have in 3D also, because it's anisotropic. You're going to get the peak somewhere, and then it's going to decay somehow. But when we use this type of crazy patterns, which this is called the tetrapode PSF, and there are studies, there are papers on how to engineer them, on how to build them, how to decide how to create them. You're going to get from the same point object, right? This is the point object. You're going to get very different patterns, which later you can use to actually learn where is your point object in 3D. Sababa? Took me a lot of time to understand that, because I heard some lectures about that, and I just couldn't understand what it is. The hard thing for me as a computer scientist is to understand that you can actually do something physical eventually. It took me a lot of time to understand that, oh yeah, you can actually build this kind of filter, right? And put it into the microscope and change your images, right? So for me, I remember that it took time, so I thought of trying to explain it in a more clear way. So the idea is as follows. Okay.
So they did it in the microscope, these images?
Yeah, but
Okay.
with filter,
So
with this filter and without this filter, right?
Okay, but there are many, so I guess there are many parameters for this filter, right?
Right.
Oh.
Right. You want, yeah. I mean, I have a paper to tell you about, right? So we want to keep a little mystery here, so you are engaged. But okay, but let's start with a given phase mask. Okay, let's start. We have a crazy pattern like this. Tetrapod. By the way, the tetrapod, if I remember correctly, is like an optimal setting for one emitter, right? For one point source. But of course, in real life, we have a lot of these, so it's more complex, and it's not the optimal setting for that. We want something better. But Okay, so, but let's start before we are looking to optimize, and Alina, you are alluding to the correct way, correctly, that we want to optimize this, right? We want to build a phase mask that will give us an optimal result for something. But before that, let's say, okay, we have one of them and let's fix it. This is what we have. Given that we have that, basically, I mean, what you could do is take this 2D low resolution image and go through a convolutional neural network or an autoencoder or whatever, right, to map it into an image that has delocalization data of the, you know, the super resolved image, and then learn a mapping and sababa, right? In principle, it's the deep storm. It's what Anatoly told us about, just with the fancy filter before, right? On the that's it. And it goes to 3D, so it works. It's cool. It's cool, but okay, I'm not very. After seeing the other papers, it's 2 years later. Who cares, right? Okay, and but it works. So here is a 2D low resolution image, and it's mapped to a 3D high resolution volume. So this is mapped to this based on these patterns that are within the image and contain information about the 3D location. The 3D location is encoded here in color. And yeah, so you have a 3D high resolution volume. So you map a 2D image to a 3D image. And then you do the localization of the points. I mean, this is just image processing, and you're fine, you're happy. So here is how it looks in 3D. I wanted to show that Rak is because it's cool. So this is what you see here. Just instead of color code, you actually go through the 3D volume. Look at it again. Yeah, good. One of the coolest things about my research domain is that you get to look at all these cool videos and pictures.
This is like a scan of the what we saw in the video is actually a scan of the different levels, right?
The different slices in 3D. Cool. Okay, so Alina, here is your question. So practically everything here, I want to make sure that I'm saying something right. Everything here is simulated, if I'm not doing a big mistake, right? It's all simulated. So if you know how to simulate every step here in the way, so given a phase mask, how an image will go through it and how it will look, right? And then going from the 3D to from the 2D result image to a 3D localization, we know we can do it from here, right? I mean, this is what I just showed you. So here is what we have. And what we are going to optimize in parallel is this, that we can refer to it as an image. It's basically optimizing an image. And here we have a network. And now we can just, simulate point objects, simulate what happens when we go through the phase mask, create a 2D simulated image, and go through mapping through a deep neural network that will generate a 3D localization. Right? I mean, this is the cool part about this paper, for me at least. And now you can optimize. I'm a little cheating here. I took this sentence from their paper, and I think they also discuss it. It's not really an optimal 3D. I mean, you are doing here a local optimization, basically. So you are doing something, you're not doing a global optimization. You're not trying all the masks in the world and trying to optimize globally, but you are starting with something and doing a local optimization. But still, I mean, you are improving your ability to generate a physical mask that you later produce and use it in your microscope. So what you see here on the upper left and in the upper right is the evolution of the phase mask and the point spread function on the dot object eventually that is as it is image. And this is the end, and you can see at the end that you can see this the patterns here, non-trivial patterns, and here are the patterns in 3D, the patterns that were learned in 3D. So no human being is able to engineer something like that. It's very complex. It's non-linear. The standard methods of engineering point spread function are not. I mean, I'm not an expert in that. I don't know how they do that exactly, but I assume that it's not the way that we see here. We see something that is completely non-trivial, and it improves the ability to localize, to do localization microscopy. So if we compare it, this is the tetrapod, which I said was, I think it's optimal for one-point source, and this is the learned low-resolution filter of, you can see that it's more dim, so there is less light here, and it's to give more contrast, because this is expected, the learned is expected to work better for more messy data, right? For more point objects, so you need to get more heterogeneity in space. At least I think, I mean, this is how I interpret that. And when we look at results, so at the top here, you see the tetrapod results in black. So this is, tetrapod is the one that we started with, right? This one, where is that? Here, this is the tetrapod. So at the top layer here, you see the results of a tetrapod versus some standard, some other algorithm, right? Matching pursuit, some standard algorithm for that. And you can see that, and I showed here two measurements. They have three, so the lateral RMSE and the axial in third dimension RMSE between detections. So it does like a global detection, then it checks the differences in space. And you can see that the tetrapod is much better than the other algorithm. And that the learned algorithm is better than the tetrapod. So it's not on the same graph because I picked it from two different places in the paper, but you can see it very clearly that it is improving, right? Now, okay, so now a little bit about the domain. So this is not, I mean, it's not a computer science story. I mean, it's not, no one cares about the, I mean, I think that the cool idea is this exactly. So the two things are optimized here. And one of them is actually the PSF, the phase mask, which is eventually a physical component, and using simulation for that. And I think that the combination of everything here makes it very cool and worthy of the high-profile publication, right? I mean, this is the contribution. If you look about it, if you look at it from computer science perspective and looking at the improvement, the results are improved. They're improved, okay, they're improved, they're even improved significantly, but it's not groundbreaking like what you see at the top here, right? It's not a huge change, it's another epsilon. And I just wanted to say that because usually if you want to, in this field, if you want to publish a method, you either need to show that you can do with it things that you couldn't do before completely, right? You measure something completely new, or you have a huge, or when you compare it to existing methods, you have a huge improvement. So you can do, again, you can do things that you couldn't do before because you didn't have the sensitivity, for example. But it's not like epsilon better, like you see in a lot of applied computer science stories, a cool algorithm and then an epsilon better, you need to show that you are a lot better. Here, I think they got some slack on the magnitude of the improvement because I think the idea is really cool. So, and once you do that, they can show some specific examples. So here is you can see one with the tetrapod. This is the experimental input. This is the reconstruction. and this is what you get in terms of localization. And you can see here that you identify, so the circles, not circles, the ellipse, because it's in 3D, so it's kind of the ellipses here are the localizations. The 3D ellipse is because of the PSF, because it's less, the PSF is more stretched in the third dimension. So you can see the gray ellipse are what you would expect, the ground truth. The green are detections of the algorithm, and the red are false positives. So you can see that with the tetrapod, you get 49 out of the 62 detections and 1 false positive. And with the learned network, I don't know, learned everything, PSF and network, you get, in terms of localization, you get 57 out of 62, which is much larger, more than here, and 2 false positives. And eventually, if we remember that eventually what we want, eventually the goal is this, right? I mean, this is what we want to measure. We don't RMC, blah, blah, blah. Okay, it's all fine, but eventually this is the output. This is where we want to get to, okay? So
the learning is done on the theropod images, right? The learning itself.
you mean the starting point?
What do we feed to the model? The theropods, right?
As an input or in general?
As their input.
I'm not sure, because if you look here, it starts, does it start with a tetrapod? Maybe. No, it starts with something a little bit different.
As I understood, it gets one picture, learned how to create the tetrapods, and then it, as you said, it basically creates the learning from the learned tetrapods to the expected picture. If you go one back,
so what you say is the starting point is the tetrapod in the training.
No, because if you go 2 slides back, before that, one before that, yes. So you said that you have a single point, right, in the green, and then you have the tetrapods, and then you said that they learn how to produce the tetrapods. This is what I understood.
No, the tetrapods is already from a different story. It was devised elsewhere. It was engineered in you know, I don't know when, maybe in 2016 or something like that.
Okay, so they just compare in the paper what they can learn from a tetrapod image but the algorithm itself works on the input of a regular 2D image, right?
So let's go back to this. I think this is the key figure here.
Yeah.
And so you have, in every machine learning, you have training, and then you have, right, and then you use it, right? Yeah. So what you have for training, you know to simulate every step in the road. So if you have a mask, you know how the optics will work, you know everything, right? You know how to build this, and this is just a network. So you just take the image and you plug it into the network and you get an output. So you optimize input to output. You know the input, you know what output you need to get, right? The localizations, and you learn all this transformation. Now, and now when you have new inputs that you haven't seen, you learn how to do the 3D localization. The question that Gil asked, whether what do we start here in the learned phase mask, whether we start with the tetrapod or start with something else, and my answer is I don't know. I mean, we need to get into the methods in order to figure it out. Hello?
May I ask another question?
Sure.
Do we need to relearn the point thread function for every individual setup of microscopes and lenses. But we are simulating presumably an existing system.
Yeah, so the question is, what are the assumptions under the data generated here? And
Yeah.
if these assumptions are changed, how it will change your ability to solve problems? I don't know, and I must admit, okay, I read the paper, but I read it, I read it, I didn't go into all the specifics. So maybe it's even discussed there. Probably it should somewhere. I didn't read the method, I just read the paper itself, and the rest is my interpretation. So maybe so I don't know. It's a very good question. I would say that if they have some strong assumptions about the way that they simulate their data, then yeah, you will need to if you go to a very different data, the way of generated data, or a very specific properties of the data that you generate, yes, you'll need to train a new network. But again, a lot of these things you find is they work well also for other data. So even if you use the tetrapod that you saw and you use it for even though it was designed for one emitter and you use it for many emitters, you still get a pretty good result, right? So you don't need to engineer every time, and this is okay, this is good for papers, but for practical reasons, you can probably use close enough, right? I could have invited you up to actually speak about this. I didn't think about it. But I thought about it only after Anatoly selected his paper, so I couldn't know it. Okay, and eventually we need to show that it's good for something. It's not just a nice trick. You can actually use it for something. And this is, in principle, okay, now we can use much higher, 3D imaging in 3D is a pain. And doing a super resolution in 3D is a pain. And now we can actually do some live imaging there. So you can do live imaging in 3D and get it to work. Now, could you do it with the tetrapod without all the fancy tricks here? And maybe it will be a little bit worse, but probably yes. But still, it gives when you build the paper, you build it, I really like the coolness factor of optimizing both parameters. And now as a reviewer, I want a review of this paper, just looking for an excuse to get it right. And it also depends on how people look. Yeah. Anyway, I shouldn't speak too much when I'm recording. Okay, so if we summarize everything here, so I spent another full hour and 8 minutes on a generative neural network for improving images, right? So the main ideas here are that we take experimental specific data and we avoid specific assumptions on our data, and we use the data to learn the mapping from a bad image to a better image, right? And this allows us to, on one hand, to reduce the cost in terms of time, in terms of space, in terms of laser power, and get a comparable result in terms of the image quality. As we saw in the last several papers, we don't need, well, of course, it's a big generalization where we're looking at very specific structures, right? You cannot say that in general, but when you know the structures and you know to simulate them well, you don't need experimental data. You can train models on simulated data, and then it still works on experimental data, which I think is a big deal. I think it's pretty cool. And 2 years ago, if you have told me that, I wouldn't believe you. 3 years ago, maybe. It also could work well out of the box. And we see all of these tools coming out now for biologists. They have no idea on anything about machine learning, that they actually can use these tools as is. And they're also parameterless. So you don't need to tune anything. It just, you just take it and use it, unless you need to retrain it. And we talked about it in several contexts, that we need to measure what we care about. And a lot of time we care about the downstream analysis. I don't care about the image itself that is generated from the phase mask. I care about the localization. I don't care about well, we just talked about the super resolution, but segmentation, the number of cells, the location of the cells. This is what we care about, and there is still a lot of work to do in these terms of defining how to measure what is on these generated images, how to measure their quality based on the task that you want to do. And we're going to have a paper on that, I think next week or the next. I think by Alon Spiegler. You're speaking about maybe next week. And Alon, after you do your thing with Yeshaya and you speak to me, I'm going to give you my reviews of this. So I was a reviewer of that paper, and so you'll be able to see what I thought about it before it came out. Another thing that I probably shouldn't say it here. Yeah. Last time I showed you that similar ideas came in computer vision like a decade before, and this is an opportunity. And if you want, I can tell you secrets on what I think are going to be the next techniques that are going to be, that will come to microscopy. I have some thoughts about that. I highly recommend you to read the perspective from Roy Erslab. I think he did a fantastic job there. And it's not new anymore. It's from I think late 2019, but it's very well written. And of course, there is my data science in cell imaging, which is like the best, right? Okay, let's take a break, 10-minute break, and we'll be back at 3:22. If you want to ask me something before, administrative or scientific, then I'm here for 2 minutes before I take a break myself. Okay. One thing that I forgot to say, if you have also, so after a student presentation, we have 5 minutes for discussion. First priority is questions about the paper. And second priority, if there are no questions or a few questions, then feedback on the presentation. Okay, what was good, what can be improved. I think in this case we had there is not much to improve anyway. So okay. One thing nice about not being able to stand on my leg is that everything comes to me. Oops, second. I'm not sharing my screen with you. Okay. So you all remember this from 2 weeks ago. This is a sketch of a cell, and the color coded is the different organelles, evronim. So what builds the cell and makes it function. And in order to understand the cell from an holistic perspective and understand how a cell works, we need to understand how its components are built together and operate together to get to a desired function. And last time I showed you a technique called the in silico labeling. 2 papers were the first that showed that this is possible. And the idea was that when you look at the phase contrast images, bright field images, so these are images that are imaged with white light, right? So without any fluorescence recorded from the specimen, from the cells. So you see here a bunch of cells in 3D, and they're imaged with just plain white light or with some, but no fluorescent here. And the question was whether there is hidden structure within these images that gives us information regarding where different structures within the cell are at. So what we would like to have is this as input, and then generate all of this as output, and see the different, and basically we would like to see how the different organelles within the cells are organized and how they're organized in one related, in spatial relation to one another, and temporal relation, when one changes, how the other changes. And if you put some drugs on your cells and you change one organelle, how it will change the interaction with the other organelles. All of these is things that we would really like to know. But one of the major limitations is that it's very hard to image in a live cell multiple organelles. And it's hard from biochemical, I mean, just plugging all these fluorescent molecules into the cell and making it right. First thing, right? Even that is difficult, and validating that and everything. And second, imaging that, because the spectral domain is interfering with one another, and it's really hard to image multiple fluorescent proteins at once. And I told you that like the best work is like 6 different organelles in the living cell, and this is a 5 years project of 2 very talented and dedicated postdocs in the best lab in the world, right? So it's not something that we can now just do everywhere. So this is what we want to get, and the way to get it is by, so we cannot have in the same living cell many proteins labeled, but we can have one. So if we're taking this one protein and we're learning a mapping, we're training a network to map from a bright field image to the protein by optimizing. So the optimization here, the loss is defined by the discrepancy between the predicted output and the ground truth of the specific fluorescent that is tied within these cells. Then we can build a model for this protein, right? And now when we do it for each protein or each organelle independently, we can get a composite representation of the cell of all of these together and start asking this question. Again, if we trust the input, and here it starts to be tricky, right? Because who says that we can trust what we generated? All of this is fake eventually. This is like the favorite time lapse. So as I showed you, the top row is the actual image data, and over time, from left to right, time here, and the rest is imaginary. The rest is fake. Everything is generated from these images, and you can see very clearly how the DNA splits and how one membrane becomes 2 membranes. So there are cells here that undergo mitosis, cell division. Okay, pretty cool. Again, the architecture here is a unit, just mapping. And what is different here? What is the distinction from what I showed here to CARE and Anna Palm and all of these guys are that the modalities are actually different here. So there is no, it's not, a lower resolution of the same thing. It's something that in principle you wouldn't expect to have the information about one molecule within the bright field image where you don't have this molecule stacked. So for me, it was really surprising that it actually can work, even can work this thing, and it does. So in one of the papers, they showed, so there are 2 papers, one from the Allen Institute, and they came also about the same time, late 2018, one from the Allen Institute of Cell Science and one from a group of UCSF and Google. And this is from the Allen Institute. We are also collaborating with them on several projects around these ideas. So what you see here are different organelles in the x-axis and the accuracy in terms of correlation of the generated images. And what you see here, a nice thing is what you see here in black, which is the upper bound. How good can you get theoretically based on the noise within the specific sample? So each sample has a different fluorescent distribution, right? You're using some fluorescent which has some background, and basically they simulated what will happen if you just add from the distribution of the background to a ground truth image, and based on that, when you have more noise or less noise, you can define the upper bound, how how good you can get theoretically with your generated images. So this is one technical aspect that I liked in this paper. And you can see that, well, some organelles are better, some organelles are worse. And now we are following up on that and trying to characterize more specifically what some properties of these in silico images. I want to go quickly over some validation results. You don't need a large data sets to train such a network. The reason is that in one network you have basically you take patches from the network, 3D patches, and in one image these images are quite large, and in one image you can take a lot of patches, right? So you can use it to train a network, and it looks pretty good from a small number of images. Another nice validation is training a model on one cell type and applying it to another cell type to see how you can generalize things here. And you train on one cell type, you're always going to get better results, but still you get reasonable results also on the other cell type. It's cool. And here I showed you this slide, but I want to show you again, because now it becomes really critical. I mean, you're not just going to miss tracking a cell, you're going to miss a cell or not miss a cell. Here, if you rely on the generated images, you want maybe to use the light, the intensity as a proxy of the, I don't know, the quantity of the protein or something. It's a problem, because if you're going to hallucinate things, if you're going to make mistakes, you might bias, or you might discover things that are completely not true. So I remind you that it all depends on what you see as training data. So what they did here, and this is part of the review that perspective reviews that I suggest you guys to read. This is the input, and then this is the input, and then the image is degraded. So it's right. I mean, you deteriorate the quality of the image, and now you want to generate it with the same idea. And you can see that you get something that looks like the ground truth, kind of. But if in your training you don't see what you're supposed to see, then you can get things that are completely different, like this, I don't know, Chinese kind of mishmash Chinese and something else. If it's Chinese, I don't even know, but text. So you can see hallucination. This is hallucination that comes from seeing training data, the test data that is different from your training data, which is, a big deal. And even on your training data, you need to be very careful. But simple things like, okay, now I put some drug on my cells, maybe it will change everything, right? Maybe it will change, it will be like looking at an A and an E without training with that, because it will change the cells to reflect new things. So that's an issue that you need to see. So I think in silico labeling, I mean, if it works and you can trust it, I would say it's a Nobel Prize, right? I mean, well, at least this is my opinion. Maybe I'm extreme, but if super resolution got a Nobel Prize, now you can actually see things that you cannot do it experimentally. You cannot measure 10 organelles at once. And here you can actually generate them. So if it's true, I think it's going to flip cell biology completely. I think there are limitations, and there are always going to be limitations to this technique. So it's probably not going to win the Nobel Prize, but I think it's very exciting that it even works. And now we need to understand what we can use it for, and what is it, the real limitations. And we need to be always very careful here. And if we find something, we need to validate it in complementary ways. For example, if we find that two organelles are, I don't know, somehow related in a special relationships that we want to study, now we can go and generate these cells with these specific two organelles and actually study the real data, right? Study a cell with what we predicted, which is also one of the cool things. As computer scientists, we usually look at the Twitter network or look at the transactions from some banking or whatever. We don't have control of that. We get the data and we need to extract what we can from it as data scientist or applied computer scientist. And as thinking as cell biologist, you can actually do an experiment that will test specific predictions that you have, which I think is a huge advantage of going into this domain. And there are a lot of questions. It's still an open-ended application. Who knows? There is a lot to do here. Okay, so this was one paper, and I want to quickly go over a second paper. So I told you the other papers that did something similar. So they showed something very similar. Fluorescent microscopy images can be predicted from transmitted light Z-stacks. This is the three-dimensional, but also the L Institute was also three-dimensional. They went to simpler readouts. So they didn't look for organelles. 19, 12, I don't remember how many different organelles in their paper, but here they used something much simpler. They just focused on the nuclei, whether the cell is live or dead, predicting cell type, things that are probably simpler in terms of the biology. But they went across different labs and different image acquisition techniques, which I think is very important. If you want to show that something works and you can show it on not on the same setting, it's much more powerful. One nice idea that they had here is multitask learning. So although they had different labels, they trained everything through the same network. And this is another idea in computer vision, machine learning. When you learn multiple tasks through the same networks that are related, you can use one task to learn about the other task, right? You can put more structure. The network learns more about the real world and it can learn better. So this is a nice idea that I think was imported into cell biology. And yeah, and they show transfer learning, so they can take the network and now you want to learn something new. You need very little data in order to train and adjust your network to something new. So this is their abstract. And so they have these pairs of bright field and different molecular markers for different things. Nuclear, dead cells versus live cells, etc. They train a network, and now when you get a bright field image, you can generate all of the panel of these images, which is very similar. This is the multitask learning. So even if you learn, you take this image and you don't have all the labels, because in each lab you image just some of the labels, still you can train your network and improve the results. Yeah, so here are training data. You can see that you don't have everything, right? Here you don't have the red channel, and well, that's it. And these are different experimental conditions. And the architecture of the networks, they use something that is called the inception modules. And I mean, I left here some simple, accessible information that you can read about it. I'll just tell you, I think that's it, yeah. So I'll just tell you the general idea. So at some point, I mean, networks were trained, deep neural networks. So people train deeper and deeper networks, right, to solve complex problems. So you get more deeper and more deeper and more deeper. And the problem was that when you go so deep, you get a problem of accumulating errors, right? It's hard to optimize this crazy model from one side to another with the backpropagation. And also that in every, when you want to use a convolution, you need to select a scale. So at what scale I'm going to analyze my data. I'm going to convolve my data in the image. And this is also problematic because if you look at, you can, especially in natural images, you can look at someone's face from very, very close or from very far away, and it will look very different. And you need to use filters that your results will improve if you can use the filters that are relevant for the specific scale of the data. So the idea of the very broad idea of inception network is instead of going deep, is going wide, and you build a panel of convolutions at different resolutions, and basically you also optimize this scaling as part of your training of your network. Okay, this is the general idea. Okay, so this is the data, and it's hard to see, but you see here a GIF, you see here a video, and the video is not time, it's the Z stack. So you can see it at the bottom, you can see it better than at the top. You see the Z stack in 3 dimensions. And here are predictions, boom. Right, so you see the input, and this is the output of, what is that? I don't know, probably neurons or something like that. And these are the neurons, rat neurons. So you have your 2 channels. This is the input, this is the ground truth, and these are the predictions. Okay, input, ground truth, prediction. Pretty good. Input, ground truth, prediction. Here are this is the again rat neurons, but now it's markers for I think live dead. You see input, ground truth, prediction. And here is the last one, I think, that I wanted to show you here something. No, this is not the one. This is something else, human iPSC neurons. So it's like stem cells for neurons. And I wanted to show you one thing. Let me see. Oh, here it is. So here is the hallucination effect. Look at the 3 here, right? Ground truth, and this is the input, ground truth, and this is the output of the network. So you see this crap? This is not there for sure, but the network imagines that. So it doesn't always work. Questions?
Yeah, I just missed. When did this paper was published? When was this paper published?
נכון.
2018?
איך לא שמתי? Late 2018.
Okay.
Okay, and Alon Spiegler is going next week is going to talk about this paper, which I think it's a cool paper from 2 perspectives, and this is what you see. This is the abstract. In principle, they show that they can do the same thing here, and there are other papers that show that they can do this in silico labeling. It's not the only one. But here they can do it in much lower resolution. This is like resolution that is more appropriate for high throughput screens. So when you want to collect a lot of data, but you can still do that. But here again, the goal here is not the localization of a specific molecule, but here when you want to do this screen high throughput imaging, and this is a topic that I'll talk about maybe even next week. I'm not sure. Then you want to get you want to collect a lot of data and then measure something, for example, number of cells, geometry of the cells, how fast they move, etc. So here in principle you can take images without annotating them and generate annotation by these networks, and then you can do tracking, segmentation, all of that. It's very hard to do it on the transmitted light images that you see here. And the other contribution in the paper is that they say, okay, but who says all of the measurements that were used in all these papers that I showed you were correlations? But I mean, is it really what we want to measure? And their answer is no, we want to measure practical measures which are related to the task. So if you want to do tracking, we need to see how tracking is affected. If you want to do segmentations and detections and detection, etc. And there are plenty of other papers coming out, and you can pick, some of them were picked, I think, with some cooler ideas and some, I mean, all of them are kind of cool in their application. Computationally, some of them are just, again, I mean, here is a network, and you map from one modality to another, and that's it. And it's a new application for a new task. And here you get, I mean, and all of them are very high profile papers. So, so in the last like 2 years, the same, actually for me, it was a little bit disappointing intellectually to see all of these papers coming out with the same idea, kind of boring. And I showed you this slide and I'm showing that again. I think that and we're seeing that now. So this was 2018. From early 2018, the technology have arrived to cell biology, right? People have, it seems that these things can work and actually improve the life of cell biologists using microscopy. But now, less than 3 years later or 3 years later, there are all these tools that are actually available now for the wide audience. You don't need to be an expert in machine learning or even in programming to actually use a neural networks that were trained for a specific task and get the results, which I think is amazing. I mean, it's really fast to, especially for a field that was so behind in terms of these ideas. Okay, so the third topic that I wanted to discuss with you is classifying cell state with deep learning. And I'm going to show you a few examples. So what we saw so far is taking a bad image and improving it, taking an image and predicting where the molecules are within this image. And now we want to ask something functional about the cell. We have a cell. Is it sick? Is it a cell from a sick patient, from a healthy patient? Can we, if it's a continuous process in time, can we pinpoint where we are in time and stuff like that, right? We want to predict something functional about the cell from its image. So one paper is this paper from 2017 that showed a methodology, and they showed it for 2 applications, showing that it's more general, right? For one of them is cell cycle, which is completely in our scope of the course, and the other is completely different. Disease progression, some disease I'll show you, but it's something completely different. And they showed that you can actually, taking an image, you can understand from a continuous, an image, a snapshot from a continuous project by exploiting the variability within the data, taking from a lot of images, you can understand where you are along a trajectory over time. So here what they did, they combined a convolutional neural network and nonlinear dimensionality reduction. I'll talk about it briefly. To enable reconstruction of the cell cycle from raw image data. So you take an image and the machine is going to tell you where is it along the cell cycle stage. So there are different stages. I'll briefly show you, but it doesn't really matter the letters exactly and the names, I mean, for us. I mean, remember it's 2017, so these things were actually quite new. Unsupervised detection of a subpopulation of dead cells. So by looking at the data, you can see an outlier and say, oh, these are actually dead cells. We can exclude them based on this kind of unsupervised specific subgroup of cells. And if we want to predict the stage in cell cycle, we can show that the supervised classification performed using neural networks perform better than using direct image features, engineered image features. Well, now, you know, now we are 4 years later. I mean, it's kind of standard, but then it was a very new thing. And then showing that this approach is not specific to one task and taking it to a completely different domain of medical imaging and showing that it works there as well. So imaging-wise, it's a relatively simple setup. So this is the machines they use is called flow cytometry. So it's a machine that takes cells like you see here, and then based on some they could be express some fluorescence, and based on some properties, the machine picks cells based on some criteria to say if there is enough fluorescence here, then pick it, and if not, then don't pick it, and then it images that one by one. So the input is one cell, and they show it in two channels. One is the bright field, and the other is the dark field image, and this is the input. And one of the shortcuts here in terms of complex problems is that it's all the pre-processing is done. So you get already a nice image of a cell because the machine does it for you, right? So you get a single cell. You don't need to do pre-processing in order to get from your image single cells. So this is a very big plus. This is a data set, so 37,000. Yeah, and what they did here, they trained a network, and they had here an optimization, a classification task of assigning for each image the stage. So you see here the stages of cell cycle: G1, S, G2, prophase, metaphase, anaphase, etc. And then using this last layer, you can use them as features. And then what they did for visualization and exploration, they did a t-SNE, which is it's a t-distributed stochastic neighbor embedding. It's basically a dimensionality reduction technique like PCA, just it's non-linear. So basically it reduces it to a low dimension where objects that are close to one another in the low dimension are expected to be close to one another in the high dimension. And it's an iterative process, and it's a stochastic iterative process. So when I'm just giving you highlights of t-SNE, I mean, so you know, just for general education, right? So if you execute t-SNE multiple times, you can get different results, right? Because it's a process that has stochasticity in it. And you can't say anything about what you can say is about when you have objects that are close to one another, they are expected to be close in the high dimensional space. That's how the optimization works. But if they are distant from one another in the low dimensional space, in the t-SNE space, it doesn't say anything about their distance in the high dimensional space. And in these terms, it's different from PCA, for example, where you get a linear transformation and your distances actually mean something in the low dimensional space. So it's a paper from 2018. Hinton, you probably know the name from his, probably the father of he was one of the crazy people still in the 80s who was playing with neural networks when no one believed in that. 80s and 90s, no one believed in this, and so this is one more of his significant contribution, and it's used widely. I mean, t-SNE is very, very useful, and now there are other non-linear dimensionality reduction technique, UMAP, for example. There are other now. They come out. So okay, and then yeah. So here are the 7 cell cycle stages, and here they collect together the G1, the S, and the G2. So this is like one class, and then you have the other 4 classes. And you can see here in bright field images. So now it's not live imaging. You just get a snapshot, right? The cells are not live, and this is not the same cell transitioning through time, but each one of these is a different cell. Okay. And here are a few markers that show you this differentiation. You can test them. You can label your cells with it, and then you can select for them, and then you can image these markers and see, oh, this cell is already in this stage or in that stage. But of course, the input for the network is the bright field and the dark field. Here is the t-SNE plot, and what you see in color coded here are the different states, the different stages of cell cycle of the cells. And I don't remember, but I assume here that the cell cycle was determined based on the markers, right? So it's kind of the ground truth, right? Yeah, because you can see it mixed here. So the ground truth is the color code, the classes. And what you see here is a nice trajectory in space that you represent the trajectory over time of the progression through the cell cycle progression. Well, it's pretty cool, I think. Any questions so far? You need to speak closer to Anatoly, closer to the microphone.
I'm sorry. I can say something about Disney that if it iterates much time, it's just overfitted and you see nice pictures always. So I don't believe to Disney.
Yeah, I mean, I would. The problem, Anatoly, you talked about the problems in methodology. I saw too many papers, or at least before peer review papers, that also after peer review, that try to argue something about the clustering of about specific clusters and the relations between them in space, which is completely crap, right? I mean, but yeah, you need to be careful about that. But I mean, if you get something like this, Anatoly, it's not by chance. I mean, you get, right? I mean, the trajectory was, it's just, basically, it's relatively an easy problem because the morphology is very clear and it's very progressive. You see, it's progressive in a very clear stage so it captures the variability within the data, captures here the trajectory of the images, right? So it's actually not surprising. And you'll see in the next hour, you're going to see other examples that use cell cycle as the task that they want to solve. So in a paper, I mean, you say, oh, I have a general technique that it's good for changing the world, blah, 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 blah. And then you go to demonstrate it for a problem. And then you pick cell cycle because it's like the easiest problem to solve, right? So, I mean, I think that this is real in terms of, because I mean, you wouldn't get this trajectory by chance. You agree, Anatoly?
Maybe I don't know.
Okay, I believe
I prefer to agree with you
until I give you the grade, right? So that.
Yeah.
Okay, so so here we get our trajectory in the Disney, and any other questions before I continue here with this paper? Okay, so here is the same data, just here, instead of putting the annotations, you look at the DNA content, right? So DNA content is kind of it's kind of a readout that is also relates, but it's a real readout, right? It also relates to the different cell cycle times. So maybe, actually, I don't remember. Maybe here it's the prediction and not the ground truth. Well, anyway, as you can see, this is a paper that I didn't read before class today. I thought that I remember it well, and I don't remember it well enough, so apologies. But anyway, the idea is it could be either one or the other. Okay, so here is the DNA content. So what the color codes that you see here is something really, something physical, right? It's not anymore something that was generated or annotated, or this is real work. And the question is, what is this group of cells? Whoever listened to what I said from the beginning should be able to answer that. No, תנחשו, you can guess.
It's or hallucination or different phase, not G1S, G2, but something else.
So it cannot be hallucination because there are no fake images here. It's not you're not generating images here. You're doing there is no, if you look at the network, there is no images in here. I mean, it's just a bunch of numbers. But if you remember that what I said at the beginning, now what you can do is go back to the data and stare at these images, right? Stare at these images. So these are cells that are taken from here, and these are cells that are taken from here. And when you look at them, you can see, oh, these are damaged cells, these are dead cells. So now, based on this visualization, you can automate the process that will tell you, okay, all the outliers do not fall in this trajectory are probably just dead cells and just exclude them. Okay, one thing that I want to highlight is that here is the first attempt to maybe do something that relates to interpretability, or what the network captures in order to make the call. It's something, it's still an open question, also in computer vision, which is much more advanced than cell biology field, but definitely in cell biology. And I'm going to show you a few other examples today and also later in this course. But this is basically shows activation patterns of different layers within the network. Well, here it's not super informative, but you can see something. I mean, what is activated based on what image? It can give you some information on what the network is learning. Not much, I agree, but again, it's early, 2017. And now comparing a deep learned network to state-of-the-art at the time boosting techniques that used engineering features and combined them together, different models together to tackle this problem. And you can see a big improvement. I mean, from 92% to 98, it's 99. It's a huge change because think about it, you can get as good as 100%. So here you have 7%, 8% to improve, and you went from 7-8% to like 1 to 2%. Okay, so it's not just epsilon; it's really big improvement. And here you get the confusion matrix, which looks pretty nice, and 7-class prediction. This is like a 5-class prediction, taking the G1, GS, and G2 together. So these are 5 classes, and this is 7-class prediction, which is not as good, but it's pretty good still.
But you can't compare it.
Huh?
It's imbalanced.
No, לא שומעים אותך.
It's imbalanced classification, and you have this G1 SG2 class with lots of samples.
Right.
So this matrix is bad.
Yeah, I agree. Right. So
you can't know with anything.
נכון, אתה צודק, נכון. You're right. And you can see here the false positive and the true positive, and probably say something from that, right? I agree that most what you get here is from the first the 3 classes that you can capture pretty well. So yeah. But still, it's a big. If you look at the improvement, it's still a big improvement. You still go from here to here, right? So this is still true.
Maybe.
Okay. Okay. And they did the same thing for retina images. There was another very famous paper, I think, from Google about the classification of this type of images, which last year one of the students picked to present in this course. Now I'm not giving you papers about medical imaging because I don't want to waste the time on that. We have more cooler things in cell biology, and the paper was terrible. Anatoly would kill it, the methodology, and it did so much noise. I mean, it was in all the newspaper around the world. When you read it, just many problems, let's say. But anyway, here it's nice because they show a disease progression. First, it was a cell cycle progression, and here it's disease progression. So people with this disease in different stages, and you can see that from the images, you can actually look at these trajectories. Okay, I'm skipping to a different paper, but I'm still taking like 5 more minutes before break. So this is a paper that I really like, and again, I mean, they looked at the trajectories of a cell cycle, right? Trajectories over time of cell cycle for fixed images, but the data was more difficult than here. First, because it wasn't what's the word? Selected. No, there is a better word, but I forgot. Anyway, selected by this flow cytometer. It wasn't like, because it's like in a real spatial context. So you had cells in spatial context, and you had to look at the individual cells and still measure them. And then there was a lot of influence also in the context. So if you were a cell in a very dense region of cells, you would behave very different in terms of how you look in your division compared to a cell that is completely alone. And they had to deal with that here, and they did really nice representation with using network properties, and they solved. I think that they solved it beautifully. One cool thing that was in this paper is that first, the last author is a former Israeli. I mean, she's now in Catherine Sloan Cancer Center in New York. Her name is Dana Peer. So again, I mean, we have the local, המקומי, הגאווה המקומית, but it's not just that. Dana Peer comes from the systems biology domain. We're going to have one talk about it, guest lecture from Tal Shai from Life Sciences in maybe a month and a half from now. The data there is for each cell, let's say single cell RNA-seq. For each cell, you have a bunch of 1,000 numbers of the RNA expression levels within each cell, which is, as a data scientist, as an applied computer scientist, is like fantastic. And sometimes I'm very jealous at this field that they have so much data per single cell measurements, and they do fantastic stuff. I really like the computation that they do in this field. But they have some other limitations. So there is, you win things, but you lose other things. The things that they don't have, and we do have. First, there is also in the techniques that they generate the numbers, the 1,000 numbers that they have. So we can have like 5 numbers, right? 5 different proteins. They have 1,000 numbers, which is much better than 5. But there is a high uncertainty regarding the numbers there, and they need to use some tricks to fill in numbers that they are not sure about, that didn't read out correctly. So this is one thing. And the other more maybe critical things are they measure. So if you look at there is DNA, RNA, and the protein, and the protein makes things, right? Protein is the function, is actually the machines that makes things, that does stuff easy in your cells. So if you look in earlier stages, if you look at the RNA, for example, you're not necessarily going to get, you're further away from the cell function, from what the cell actually does. And the other thing is the space and time. So in microscopy, we have the information, the spatial information, and we can have temporal information about what's going on. So we have the context in space, and we have the temporal information and that field of single cell RNA-seq, which again is fantastic, do not have that information. Spatial context they sometimes have in some applications. Anyway, the cool thing about that is that they took tools that they already developed for the single cell RNA-seq, and they imported that, and the tools are looking at graph similarities, so building like local networks that are based on local similarities between cells in these high dimensions, and then building from that networks and trajectories within these networks. And they took this idea and applied it into microscopy data, and I really like it. I really like how they did it, and they're taking the idea from another domain in here and showing that it actually makes sense. But eventually in the applications, they did something very similar. They looked at the trajectory in space of fixed cells within a sample, and they showed some, I mean, I probably am not going to show this paper, but if someone wants to pick it as a paper to present, you can go ahead. Okay, let's take a 10-minute break, and get back at 4:19 for the last hour. I'm here for a minute if someone wants to say something or talk to me. Can I start? We're getting listen פחות ופחות. Samuel, are you with us? Otherwise, I'll speak Hebrew.
No, I'm sorry. I'm eating, so I didn't feel it was polite to eat in the class. So that's why.
No, it's fine. It's fine. I like to see you eat. I also eat, so you feel more comfortable. Okay.
Thank you.
And drink. Okay. Let's move on. Okay, so what I want to show you now is another paper that does something similar, but it did also a step ahead in terms of interpretability, I think. So the idea here is again, predicting and visualizing. So here again, visualizing is not just doing a classification, but also using generative networks in order to see how things progress over time. From snapshot images of single cells using autoencoders and optimal transport. And I want to really quickly just to highlight, and this is also a paper that if someone wants to present more deeply, I'm happy to, it's open. I think it's there I think I included in the list today. I just want to go over one idea here. So basically the question here is you have these cells and they are, okay, so you have your cells that are fixed, right? You have snapshots. Fixed means they're not live images, you have a snapshot of them in time. And there is a dynamic process, which is their differentiation. So the days are days from starting a differentiation process and progressing through it. And then they use an autoencoder to create a representation of each of these images and use optimal transport to predict trajectories in these spaces. And then they can also take an image and artificially shift it into later stages of differentiation. So what you see here is you can see the fibroblasts, the cells at day one. And in order to interpret what is changing through the differentiation, you can see the predicted change in the developmental trajectory. Through, I don't remember exactly what day it was, but in principle, you can see the changes. And from that, you can start understanding a little bit about what has changed within a cell along its differentiation trajectory. Now, one thing that you can see it very clearly, and I'm not saying it, I'm saying it with a purpose because the next story that you're going to say it's not going to happen, is that you get your still in an unsupervised manner, and all of these pseudo time or not, I'm not sure if all, but at least the ones that I've seen, these pseudo time techniques. Pseudo, it means, you don't have the real time. So you have like a trajectory based on your data, the pseudo time. This is unsupervised, right? So this is just from the visualization. So just from the dimensionality reduction, you get these trajectories that represent the progression through time. No, it's not in the next paper, but the one after that, I think. So we're talking about differentiation. Let's take another step at looking at differentiation. And this time we're going to look at early prediction in time of future differentiation. Now, in terms of application, I showed you a few papers now. And in terms of application, it's about the cell cycle from these flow cytometry images and again cell cycle and then what was that, and the differentiation progression. But here it's really, can you predict early in terms of generations? So can you take a cell in its early stages before it will take a few more generations until it commits its final functional destination. So stem cells that take geza and differentiate what's the word? Differentiate? No, in Hebrew. מתמיינים, שהתאים מתמיינים, and the cell differentiate, התאים מתמיינים, יכולים, they can differentiate to skin cells, to muscle cells, to bone, to neurons in your brain. So the stem cells can take all of these roles. And one Nobel Prize is for reverse engineering them, so taking them the other way, not reverse engineering, but taking them I forget all the word, but it's kind of redifferentiation. There is a better word for that, which I forgot, which is another prize, Nobel Prize. They show that you can add 4 components and you can take a differentiated cells and bring it back to a stem cell that can still make all the choices. The question about how a cell decides to differentiate and how the context, how other cells are involved in this decision of a cell to differentiate are big questions. Because think about it, it's the same cell, the same stem cell, tag geza, that decides to either be a bone, right, or a cell in your brain, which is crazy. And it's all context dependent. It all depends on where the cell is at, what it feels from the environment and from its neighbors. And there is a lot of studies about that, and we're trying also in the lab to do some work on this broad and very important question. Anyway, I wanted to highlight that because this is really a task that is impressive, I think, and the others are fine, but are not, are not mind blowing. I mean, it's nice that the technique is nice, and they brought a new technique to the field, which is very important. But here also the application is really cool, I think. So the ground truth here is that you have markers that can be used to say, okay, when this marker lights up, it means that the cell has committed to a fate, committed to be whatever. And the question is how early you can make this prediction. So here's the data. They had 3 experiments, and they tracked the cells. They did what is called lineage tracking. Lineage is shoshelet. This is so you need to track the cells and be super accurate, because once you do a mistake, you lost all the rest of the trajectory and all the lineage. If you lose one, and this is a lot of work. You don't do it completely automatically. Never. I mean, people develop algorithms for lineage tracking, but eventually people that do that always do manual validation of everything there, because you cannot do mistakes. And they eventually track 6,000 cells for 8 days, 2.4 million images. And the question is, again, I mean, you have markers for different differentiation fates, and you want to see in early stages, in early generations, and ask and make a prediction. So this is the experiment. We have mice, cells, long-term high throughput imaging, microscopy, tracking of the lineages. So these are the lineages, right? This is how the Etzeilana Yochasin, Shushelet, what you do in 7th grade, that you do it with your grandfather, and etc. So here they do with the actual cells. And then there is the fate, which is these are becoming blue and these are becoming red. And the question is, would you know that they are going to become red already in these early stages, based on, I don't know, some morphology, I don't know what, right? But whether there is information hidden. By staring at the cells, the answer is not clear. I mean, you don't know how you would make this call. So this is the data. You can see the tracking the cells over time. This is 26 hours. That was the experiment work for more. And here is what you see. So what you see here, every time that you see here on the bottom where you see a kind of a black trajectory, right? It's a generation. Okay, so you have a generation and you have in this case, the marker is cell size. So you see when the cell divides, it becomes smaller, right? It becomes smaller and then it grows. They grow, and grow, right? I mean, these are the generations. And 0 is when the marker lights up. And you can see this blue marker lights up here, which means that, okay, the cell is committed. This CD1632 means some commitment to something which I have no idea what it is, and I don't care about in the context of this course, definitely. And so from here, the cell is committed. And the question, can we make this prediction in early generations based on how the cell looks like, right? And using temporal information as well. And here is
Assaf, can I ask a question?
Yeah, just one more sentence. So you saw we had here a blue fade and a red fade, and you can see the same here. So you can see another trajectory, right? Other lineage tracing of cells. This is the red fade. And I mean, by staring at the images, it's really, I mean, it's hard to say what, if you look at early stages, even at late stages, but definitely early stages, it's really hard to say what differentiates these cells from that cells. But these become red and this becomes blue. Yes, Samuel.
So they just took like a million snapshots of the whole time frame of the experiment and then mapped the whole thing?
It's not snapshots, it's actually movies. This is the data.
Okay.
Live imaging.
Okay.
You cannot do lineage tracking without live imaging.
Okay. All right, that was my question. Thank you.
I had the chance to grab a few bites. So here is the network architecture, which is not very exciting. I mean, just a simple network. Maybe an interesting component here is they add here some information that relates to the displacement, to the motion of the cell, not only its appearance. Okay, so you have this information, and then you can build also you can build a model that is involving time information. So this is what you see here, and I don't want to go into the details because there are now much better ways to do these things, to model these things in time, and also I don't remember right now exactly the details, but it doesn't matter for the purpose of the points that I want to show you. And you can read, I put here a few information about using networks for temporal information and what are the limitations of recurrent neural networks and long short-term memory, which are the methods that are the networks that are shown here. Okay, so identifying and predicting lineage for morphology and displacement. So now we use this information, and we want to say in early generations whether we can predict what is going to be the outcome. And what you can see here very nicely, and here, okay, this is important, and I brought it here because a lot of times this is not a bottleneck. This is a subtle point. This is a weak point. Anatoly looking for problems in papers. So this is a very common problem of mixing together training and testing sets. And in microscopy, it's not only mixing together from the same experiment, the same cell, which is, I mean, you're not supposed to do that, and most people do not do that. But sometimes you mix other things that you're not supposed to use. I'll give you 2 examples from other papers, and then we can talk about what they did here. I have one paper that I really like about the involvement of the tumor microenvironment on the aggressiveness of a tumor. So basically what they did, they didn't image the tumor cells at all. They just imaged the composition of the environment around the tumor cells within a living mice. Living mice, I think, or maybe dead mice. I think a living mice. And from that they made prediction on how aggressive the cancer is going to be, whatever. And they showed, basically that, and they did a really nice work. I really like this paper. They showed that you don't need to even look. I mean, there is information in the environment, so that it's not only the tumor cells, but also how they communicate and how the environment changes in response to the interaction with the tumor cells to make predictions, which is cool to know, but it's not relevant to what I want to tell you. What I want to tell you is that in order to do the training testing partition, they have, for each mice, they took multiple windows into the tumor from different places. Now, in order to do the train and test, they took each window was an independent observation. They took all the mice, all the windows together, and they mixed everything together, and then they took like 20% as the test set and training set with the rest, something like that, right? Now, what's the problem here? You are biased by what you want to do. You want to take a mouse and take it out, right? You don't want to overfit your models to mice that you already seen in your training data, right? So the right way to do that, and it's a really fantastic paper. I really like it. I think that what they found is real, and they're also in a very good journal, etc. Cited many times, but it's but basically you need to take the whole mouse, take it out, train a model, and then test it on your mouse in order to convince that you are independent. I'll tell you another story. I was a reviewer for a paper in probably the highest journals that you could think of for IVF, or in vitro fertilization project. And they had their multiple hospitals. So think reduction from a hospital to a mouse, and they did it again. I mean, they mixed all the hospitals together. They maybe overfitted to the hospital, and then they made predictions, which I mean, it's okay. I mean, you just need to show that to do the right partition in the training testing without introducing biases into your system. You want to make it as independent as possible, so we can trust the results and not think that it's an overfitting to an hospital. So you can now go to a different hospital and be more confident about the ability to predict something real and not just, okay? So I spent like 5 minutes on that because I think it's very important, and people, probably you guys don't do it, but people who come from other domains and want to do machine learning, a lot of times they do these mistakes. And in cell biology, it can come from different directions, the overfitting. So for example, you can have, in this case, maybe the experiment is the bias, all right? Maybe the experiment is the overfitting, because maybe, cells feel differently. You take cells from the freezer, one time they feel better and sometimes they feel worse. Or from the mice, these are from different mice, the cells, every time that they took in this project. So they did it nicely. They did the training and testing separately, and this is why I kept this figure, although it's kind of a nitty-picky point. I think it is important to say that. Okay, and now after I kishkashti for so long, we can look at the area under the curve, and we can see these are the annotations. So when you have the ground truth, when you know already the outcome, you get area under the curve, which are pretty nice, 0.9, 0.8. I mean, think about it, because these are your data. Your data are the bright field images, not the markers, right? I mean, it's not trivial at all. And this is actually pretty high, I would say. But if you go even further, even 3 days to the past before the commitment was done, so not 3 days, 3 days or generations? Generations, sorry. 3 generations in the past, right? So only the great grandson, right, or something like that, is going to be the ones that actually commit. And still, from the great grandma or grandpa, you can already understand what is going to be the outcome of the cell, above random, definitely. Which I think is cool. And now for the best papers that I'm going to show in the course. Oh, it's mine. No, well, but it is a cool paper. Of course, I'm biased here, and you should take it with a grain of salt. And please be critique. This is a paper that is out in bioRxiv, so it's out for actually almost a full year now, I would say. And actually, we got, so formally it's not accepted, but it is accepted. So it's kind of it's done. It is accepted, but now it's the final fixing it to the format of the journal, and that's it. Maybe I'll do it today or tomorrow. I want to highlight here in this paper the main problem that we try to pitch here is ways to deal with interpretability. So neural networks are very powerful at identifying patterns within the data, complex patterns. And basically, if there is a pattern within the data, your network will capture that. Did you hear my cat? Say hello to Henry. Okay, so, but we get an outcome and we have no idea where it came from. Well, I mean, what was the reason that the network decided to decide it this way rather than something else? And this is in cell biology and in medicine also, it's critical. Because if you come to a medical doctor and tells them, okay, the machine tells me that this person is going to die or not going to die or whatever, they're going to say, based on what? And if you go to a biologist and say, this cell is going to whatever, differentiate or not differentiate, the first question is based on what? And the based on what, even in biology, when you're not going to necessarily save lives, maybe in the longer term, but the question is still very important, super important, critical, because eventually as biologists, we want to understand what is driving things. So if we know what is the reasons that our network thought that something that we cannot see by eye is causing something else, we can then try to understand how to perturb it, how to perform an experiment that will change something around what we discovered, and we'll see whether we still get the outcome or not get the outcome, so we can test experimentally and learn about what is the cause-effect relationships here in terms of the cell function. Okay? Okay, so the question that we ask is, can we predict cancer cell functional states from live label-free cell images? So, the cancer cell functional states, I'm going to tell you exactly what I mean in a second. Live, it means that we have dynamics if we need, we have time. Label-free, you know that we're not imaging any fluorescent protein within ourselves. And now I remind you that we have, actually when we started this project, we didn't know that, but now we know that phase contrast microscopy contains a lot of information also about the molecular organization within it, hidden. So we might think that there is hidden patterns within it that describe the functional state of a cell. And specifically, we started it with label-free based on the assumption that the texture within these images have some information that is hidden from the human eye. This is one thing. And the other thing is just practical, because we took cancer cells. I'll tell you very soon about what cells we took. We took cancer cells directly from patients, which are and then put in the. I'll tell you immediately what is the experiment. But the cells were super sensitive, so we couldn't really interfere with them and do a lot of putting in them fluorescent markers, blah blah blah. You can't. You want to just image the cells in their natural state. Why melanoma? So we picked melanoma because melanoma is a disease that is known to have a very high genetic heterogeneity, which means that you have different parts of your genes that are disrupted, right? That are mutated. So there are different mutations that are eventually converging to the same cell function, which is leaving a primary tumor, invading to the bloodstream, surviving in the bloodstream, going out, extravallating in some distant organ, forming a tumor, and eventually killing the patient, right? I mean, there are very different genomic signatures that lead eventually to the same functional state. And this is why we thought, hey, maybe by looking at the cells, we can actually get information where a lot of other studies tried to use genomics and couldn't do a very good job. And I'm going to show you some of this data in a minute. First, about the system, and this is a project that I started in my postdoc. in the med school that I did my postdoc in Dallas. And again, it's a long project. It just came out, just going to come out this year. And we took advantage of a very cool experimental system that was developed by the lab next door, the Morrison lab. And what they did, they took stage 3 melanoma. Melanoma is skin cancer. Stage 3, it means that the cells are already, they're not in the main tumor, in the, in the skin, but they're already in the lymph nodes. They already did some, they're already aggressive. It's already an aggressive cancer, right? Stage 3. And the question is whether it's going to progress to stage 4 or not under treatment. And stage 4, it's like metastatic all over the place. And stage 4 is, until several years ago, it was a death sentence. I mean, you couldn't get out of it. Now, for some of the patients, there are actually kind of very amazing treatments that work for some of the patients. But one question is, can we predict that based on the images? And so, but the experiment was taking the cells from the lymph nodes, xenograph them into mice, so injecting them into mice, and seeing what happens to the mice. And they found in this paper from 2012, almost 10 years ago, that there is a correlation in the outcome. So if the person is unfortunate to have aggressive melanoma, the mice also encounter something similar. And this gives a very powerful and physiological relevant system to study melanoma, because most of the studies on cancer from the perspective of cell biology, and definitely computational side, is taking cells that are what is called cell lines, which are in culture for years and years and generation of cells, and they undergo some, they are treated with some chemical treatments that makes them easy to handle. So we can grow them in the lab, they can stay alive forever, etc. They can be very, when you can do a lot of stuff with them and they are still feeling happy, but maybe they are very different from cells that you take from fresh tumors. So this gives us a very powerful setting where we can take the cells from the mice that are correlated from the mice tumor, right? You know what is going to happen to what happened to the humans that had these cells, and then you can image them. And I mean, this is a 2012 paper. And from that point, the Morrison lab and other labs are also trying to understand, okay, what makes a cell what makes one tumor super aggressive compared to another tumor? And what I show here is all kinds of genomics assessments, and also there are some cell biological assessments. Anyway, it's really hard, and you cannot really make a clear distinction. And I want to explain to you why, and I want to explain in one word the central dogma of biology. So genome goes to DNA, goes to RNA, goes to protein. So genome, transcriptome, proteome, and the proteins are signaling to one another, and it's a network, right, that they activate each other and works together, which eventually leads to a cell state and function, which leads to the outcome. Now, it's not that simple. It's not a linear trajectory, because in real world, it looks like this, right? So there is feedback that goes back, and everything is much more complex. And this is at a single cell level, but if you add the environment, it's even more complex. If you add other things that happen in the environment, it's even more. So ideally, it's not very surprising that if we look here at the top, it's going to be hard to figure out something that maybe looking here at the cell state and function, what the cells actually do, is more informative in terms of what will happen eventually to the human being, right? I mean, eventually it happens at the functional level. So it's, again, there are trade-offs. Therefore, for some things, genomics is great, and some cancers are solved thanks to the genomics. And also in melanoma, there are some treatments that are based on genomics which are very good, but in some cases where the genomic heterogeneity is high, and when you have a lot of other effects, it could cause something like this. Okay, so all of this introduction is just to tell you that we need functional readouts to stratify melanoma. And here are the experiments. So what you see here are single cells. Each guy here is a single cell over time. And one of them is a high metastatic melanoma, and the other cells come from a patient of a low metastatic melanoma. And the question, can we make predictions? And of course, from staring at these images, you cannot say anything. And even when looking at cells, I mean, okay, these cells are relatively different, but which of them is high, which of them is low, which not, and definitely it's not representative. The cells are very different from one another. Looking at parameters that are standard for this type of analysis, like trajectories, or the morphology of the cell, the shape of the cell, and stuff like that, didn't find anything. I mean, it just didn't work. You cannot find anything from that. But we were still hopeful that these ideas of using generative networks, because we know that the phase contrast images contain so much information about the internal state of the cell, maybe it will be informative regarding the metastatic efficiency. So we developed an image analysis pipeline where we, in images sequences like the experiment like I showed you here, we identified single cells, we tracked them over time, and now the atomic units that we're going to use are these images over time of a single cell. So these are the inputs. And what we did now is generate a unsupervised representation of the cell appearance. And this is, this is an autoencoder, right? It's not a unit, but it's an autoencoder. So I want to go quickly through this autoencoding. So you have the encoder part, which takes an image and compresses it into, in this case, a 56-dimensional vector. Then you have the decoder that takes the 56-dimensional vector and decodes this into a full-blown image. The optimization is minimizing the discrepancy between the reconstructed image and the input image, which means that in here, we have a good representation, because from the 56 numbers you can generate the full image. It means that we have here a good representation of the cell appearance. The part that you see up here is an adversarial component, and the adversarial component is used as a regularization term to optimize the ability of the network to distinguish between representations, latent vectors that came from real experimental data, compared to vectors that are just random, just come from another distribution. Okay, so it's kind of a regularization that helps the network become more smooth and more focused in representing cell appearance. Now, we put all the cells that we had into this network, and all the cells, it means high metastatic, low metastatic. We took cell lines. I told you that there are cell lines that are have been in culture. So these are cells that came, PDXs are cells that came from human patients through mice. Cell lines are cells that are not for many years are now in culture. They look differently, they behave differently. We took melanocytes, which are healthy cells. Everything goes into the network. Why? Because we want to learn a general representation of how a melanoma cell looks like, right? There is no annotations here. It's completely unsupervised in terms of the labels of high or low metastatic efficiency. We just want to get a good representation of the cell appearance. That's it. And later, what we're going to do in the next step is going to use the representation for a supervised step when we're going to optimize whether it's a high or low metastatic efficiency. And in this case, another advantage here, that here we use all our data. Later, when we want to compare specific classes, we might look at small classes. So for example, melanocyte data, the skin cells that are okay, that are not cancer, as they are, we didn't have a lot of data from them, so we couldn't use them to train a supervised network. But once we have a representation, we can use standard machine learning techniques to do classification. So here is the network training, the reconstruction, and actually there are better networks to generate better reconstruction, and we have one of these also. But actually, it was better to use a network that is not reconstructing the fine details of the cells for the tasks that we wanted it eventually to achieve. So we didn't need the resolution, a very high resolution. We prefer to, I mean, it worked better to have a more. So you see here the evolution, right? It starts from something and it gets, or here, for example, at the bottom, you can see it starts from this blob, and through time, it becomes an elongated cell, okay? So this is the generative nature of the network. And now, once we have that, we can take this unsupervised representation, the latent vector representation. Again, it's unsupervised. It doesn't know anything about the cell that it's looking at. Give it a cell, get a 56-dimensional representation, and then go and with this representation, go to whatever machine learning technique, supervised machine learning technique we want. Here we use LDA, linear discriminative analysis. It would work also for another classifier. I mean, it doesn't matter. And then once we do it, we can discriminate between high and low metastatic efficiency. Now, talking about the mice and about the importance of cross-validation and all of that, doing the right train and test comparison, well, we were very careful about that. First, what you see here, we have patients. For each patient, we had multiple days of imaging. We had a lot of cells for every patient. We trained different models. Each model was blind to the patients that was a test, right? So we did rounds of training, testing. Each time we took out a full patient, all the cells from a patient, and then used it as a test set. So this was one thing. And the other thing, because we did in the same day, we imaged cells from different patients, we did another round where we took the data set of a specific patient in a specific day, took it out, and took out all the data from the cell patient and all the data from the same day of imaging, and then train the model and try to predict it. It is another advantage of the ways that we analyzed our data of the unsupervised and then of supervised layer, because not layer, a machine, not a neural network layer, but analysis layer, because we could do all these many rounds of training and testing, and we didn't need to train like thousands of networks, right? Okay, so we could discriminate between a high and low metastatic efficiency cell, and it was very robust. So here we took images and we deteriorated them. So we added the blur, we changed the illumination artificially in the computer, and you can still see that the classification is okay. 50% is random. Remember that the classification here, the area under the curve here, is at the single cell resolution. Now, for one patient, we have many cells, right? So it's enough to have 0.7, and melanoma is super heterogeneous. So each cell is going to be very different from, the cells are very different from one another. So 0.7 is enough. I mean, at the patient level, we do perfect classification. Each patient, we knew exactly whether it was a high metastatic or a low metastatic patient. So this is also an important point to say, because eventually we are doing the classification at the population level, but we are measuring here at the single cell level. So it was very robust. And yeah, maybe I'll 2 more minutes, and then I'll stop with a big question mark that I'll continue next week. Okay, so that's fine. I mean, it works, it's robust, we're happy, but it's all computational. How would we validate what we get? I mean, what cool validation can we do? So I told you PDXs, it's what comes from the human patient. Cell lines, it was what is in culture for many years. It's not, you know, it haven't seen a human in tens of years. It's very different, it's treated very differently. And when you do classification between PDXs and cell lines, you can see that we very easily we distinguish between them. So they look differently from one another, okay? So what we did now, we took the classifier that we trained on the PDXs, on the human melanoma, on the high versus low, and we used it. And we have different classifiers because each one we create a different classifier for each patient. These are the patients that we have. And again, we wanted to be blind to the patient, so we have 7 different classifiers. And then we took our cell lines, and for each one we made prediction whether it's a high or a low metastatic efficient cell line. Now remember that the models that we trained have never seen a cell line, and cell lines are different from PDXs. So the question is, whether the properties that the network captured are still encoded within the cell line, the properties that are related to metastatic efficiency. And to test that, we took the A375, what we predicted as high metastatic efficiency. We took the MB3, what we predicted as low metastatic efficiency. We put them into mice, so we don't know whether they are high or low metastatic efficiency of the cell lines. We just have our computational prediction. We plug them into the mice, and voila, the primary tumor is similar. You don't have a lot of change in the main tumor. Actually, if you look carefully, even the low metastatic is going faster than the high metastatic. But when you look at remote metastasis, what is eventually determining the metastatic efficiency, you see very clearly that the A375, the one that was predicted as high, is really high. And this is pretty, I mean, I'm talking about myself, but it's pretty amazing because the network have never seen a cell line, and it still can make prediction in cell lines regarding metastatic efficiency, which I think is very cool. Okay, and I'll finish with the following slide. Okay, so I want to be a cell biologist, right? Cell biology is not enough to show that the method works. We want to understand why. Why the classifier thinks that this is the high and this is the low? What is the cellular properties? What did it capture in the image to make the call? So one thing that I did is just look at images, rank them by what the classifier thinks, and stare at them. And I stared at panels like this. So this is high metastatic and this is low metastatic. And I stared, and I couldn't see anything. And why is that? Because the variability there is a lot of variability within the data, but it's not like cell cycle where the variability within the data is what you see in an unsupervised manner. You see an image, you can say this cell is dividing more progressed or less progressed. Here, there is a lot of variability within the data, and only a very small portion of it is determinant of the metastatic efficiency. And you cannot see that by eye, and I'm going to stop here, and next week I'm going to tell you how we interpret this mystery. שאלות? סליחה שגנבתי לכם 3 דקות. Questions?
Only one small thing. Again, what you put into the classification model is not the image itself; it's the middle of the middle vector, right? The middle layer in the okay.
In the autoencoder, exactly.
Yeah.
So this is a latent representation of the cell appearance.
Latent vector. Okay.
Yeah, and now I'm giving myself a spoiler for next week. The one cool thing about the autoencoders is they have an encoder part and they have a decoder part. And let's leave that for next week. Hello? More questions? Later.